Let's now talk about lysogenic, also known as temperate phages, a really interesting category of life form. This image here is showing the life cycle or lifestyle or replication style of a lysogenic phage. So here is the host bacterial cell, and here is a phage, and it is attached, and it has injected its phage DNA. And then what's happening next is rather than immediately taking over the cell, multiplying, blowing the cell up and going off to a new host, that sort of like simple evil lifestyle of the lytic phage, what's happening is this phage DNA is actually integrated into the host cell genome. So it's become part of the host cell genome. And in that form, it is referred to not as a phage, but as a prophage. So it's simply a genetic element within the chromosome of the host cell when it is like this. And it can persist there for many rounds of cell division. So each time this cell goes through binary fission and divides, then that phage DNA is copied along with the rest of the chromosome. And so in this way, that phage DNA can be propagated through a large community of microbial cells. You can go from having this one quote unquote infected phage to, thousands or millions very, very quickly. Another thing that's really interesting is that while the phage is in this prophage state, it's not necessarily an inert genetic element. It's not just doing nothing. It's actually contributing to the phenotype of the host cell. And I'll give you some kind of scary examples of that in a moment, but it's actually influencing the ecology and biology of the host cell. So this is a pretty important aspect of bacterial genetics, is this interaction that they can have with lysogenic phages. So back to the simple. Here we go, and we're replicating along and replicating along and replicating along. And in fact, sometimes the phage DNA can just really permanently be stuck in the bacterial genome and just be left over. But for most of these, in the basic sense of this life cycle or history, what will happen is at some point in time, some environmental cue will trigger this prophage. It's like a wake-up call. It's like a zombie switch, right? And this signal will go out, and the signal can be UV light or chemical change or starvation or temperature shock. There are a lot of different possible signals that can affect this. And what will happen is the prophage will stop being inert and it will start being lytic. And so that's what this arrow is implying here. This prophage has been woken up. It will go off and it will actually take over the host cell. It'll bust up the rest of the host cell chromosome. It'll excise and it'll take on its own viral sort of identity again, and it'll make the host cell replicate it, and it'll reform actual phages, and it'll go off and it'll blow up the host cell as it does, and it will infect new host cells. So it switches back into a lytic type mode of life. So I said I would give you some examples of how phage in their prophage form can influence host cell, bacterial cell biology. And here's an example from medical microbiology. These are 1, 2, 3, 4, 5 different bacteria. They're all pathogenic bacteria. They're all bacteria that can cause some pretty harmful disease in humans. And here are the diseases on the right that they can cause. Vibrio cholerae can cause cholera. E. coli, some forms of it, of course. Some are harmless, but some forms can cause hemorrhagic diarrhea. That's blood-containing diarrhea. Clostridium botulinum can cause botulism, an extremely dangerous form of food poisoning. And Corynebacterium diphtheria can cause diphtheria, which can kill you, especially if you're young, from the shock you go into due to the fever. Streptococcus pyogenes can cause what's called scarlet fever, a very dangerous strep infection that used to be very common a number of decades ago, less common now. All of these dangerous pathogens, one of the things that makes them really dangerous, one of their virulence factors, are the toxins that they make, and they are listed here. Cholera toxin, that should just be an A there, no E. Shiga toxin, botulinum toxin, one of the most dangerous substances on Earth, in fact. Extremely, extremely dangerous. Tiny, tiny amounts can kill an entire city of humans, basically. Diphtheria toxin, and what's called erythogenic toxins, harmful to red blood cells. The genes that encode these toxins are not bacterial in origin. They are phage in origin. So Vibrio cholerae got this like deadly superpower of making cholera toxin when it was infected by a phage called the CTX phage. Clostridium botulinum got the superpower of being able to make botulinum toxin when it was infected by a set of what are called clostridial phages, and so on. So there are less harmful examples of this, but those are some interesting examples of how lysogenic phage can influence host cell biology, and by host cell I mean bacterial cell biology, in ways that have relevance to humans. So now let's switch back to the focus on horizontal gene transfer and ask the question, how can a lysogenic phage result in horizontal gene transfer in the bacterial world? And in other words, how can they participate in what is called transduction? And this cartoon is showing that. The kind of transduction that the lysogenic phage usually do is called specialized transduction. And it is shown here on the left. So here is a bacterial host cell. And in here in like the darker green, it's a little bit hard to see, is phage DNA in the prophage form. So this has been infected with a lysogenic phage already. Okay? Highlighted in red is a bacterial gene of interest, the galactose gene. And it's highlighted in red just so that we can track it through this figure. Okay, so this is a bacterial host gene. Here's the phage DNA in green, darker green. So induction is the name of the event that occurs when an environmental cue tells a prophage to wake up and act like a lytic phage. Okay, wake up and get out of the host cell. I just mentioned that. So normally what happens is the prophage DNA will go through something called excision, and it will excise, making these circular forms really cleanly. Okay, notice that all of this DNA, which has been replicated and is about to form new phage, these are phage down here. The authors have drawn them really simply. They haven't put their tails on them. They're all clean, normal, lysogenic phage going off to infect. This is what happens most of the time, okay, is clean excision or coming out of the prophage DNA from the bacterial host genome, and then a replication event, a lysis event, and off to infect again. Once in a while, though, this excision stage does not go correctly, and a bacterial gene located right next to the prophage DNA accidentally goes into the excised DNA that is then replicated, packaged up in phage, and released into the nearby environment. And we can track that in red, okay? That red bit is a host gene. Because it was located right next to the prophage, and only because it was located right next to the prophage, because of that location, that bit of bacterial DNA was integrated into the new phage genomes, packaged up and released. Now these can go off, infect new bacterial host cells, and then they can be replicated, and those new bacterial host cells are the recipients of this donor bacterial cell's little galactose gene. Why is this called specialized? Because only genes adjacent or beside the prophage site can go through specialized transduction. It's a rare event, but because of the replication of the phage before it goes off, before it blows up the host cell and goes off and infects new phage, you go from a little to a lot, like a lot, thousands and thousands, really, really quickly. And then that host cell DNA this little red bit, gets the opportunity of integrating into a lot of new recipient cell genomes. And so this is in fact like a pretty efficient way to transduce DNA, or to move it through transduction, the horizontal gene transfer act called transduction. Okay, so that's a little bit about transduction. It's a lot about transduction. And in the goals, I gave you the general things that you should know and you should have integrated into your study notes. I recommend diagrams as well. And if you actually understand this, then you'll be able to apply that knowledge to these questions, which ask you to take it to the level of application, a higher level of learning than simply memorization. So defining the term prophage and stating whether it's a term relevant to lytic or lysogenic, I will call that pretty much memorization. Do bacteriophage that conduct lytic lifestyles use specialized or general transduction? Do bacteriophage that conduct lysogenic lifestyles use specialized or general transduction? That's application level. Do both lytic and lysogenic bacteriophage lyse cells? That's a little bit of a more complex question, and to answer it properly, you'd have to give a little bit of detail on the life cycle of lysogenic bacteria. That's not a simple yes-no question. This question, can any gene in the host cell for a lysogenic phage be transferred by specialized transduction to a new host? That's a pretty simple question if you were listening, taking notes, and keeping up with this lecture, and also doing the reading that I recommend. And finally, can any gene in the host cell for a lytic phage be transferred to a new host? That requires a little bit of thinking too. And with that, I conclude this lecture set on horizontal gene transfer and in bacterial/prokaryotic genetics.